Hi everyone, my name is Marilena Pagano and I'm the Application Manager at Hellma. Today, I would like to welcome you to our webinar with R&D Manager Noémie Caillol on the topic Exploring New Methods to Evaluate Diastereomer Formation in APR Intermediate Synthesis, NIR, IR, Raman and UV spectroscopy as an alternative to HPLC in real-time monitoring. Today's expert, Noémie Caillol, is highly skilled PAT specialist with extensive expertise in advanced spectroscopy techniques, including Raman and NIR, alongside cutting-edge chemometrics for sophisticated data analysis. Since 2016, she is working as an R&D manager for spectral analysis and chemometrics at Axel'One. In her role, she is responsible for the online monitoring of chemical processes and helps industries optimize their operations through advanced analytical technologies. We are glad she found the time to share her experience with us today. Welcome, Noémie.
Welcome. Hello. Thank you very much.
You're welcome.
So hello, everybody. I'm very happy to be able to present to you this work that was done mainly by Ma-Elaine Fallow, a student of mine last summer. And so where we have been exploring new methods to evaluate diastereomer formation in API intermediate synthesis. So the idea is we have a lot of spectroscopy possible to try to use as an alternative to HPLC, so to be able to gain information in real time for monitoring the process. So the outline of the presentation will be a very short presentation of the companies this work was done for and by, and an introduction on really the products we're going to be studying, the reaction and the objectives, the methodology, results, and of course, conclusions and discussions. So Servier is an international pharmaceutical company whose headquarters is in France. They've been doing medicines for a long time now, and they are really focused in 5 therapeutic categories, including oncology, cardiovascular, immune inflammatory, neuroscience, and diabetes. So the need that was presented to us in Axel'One was really came from oil industry. So there are really a production plant that has been working for Servier for a long time in producing the APIs for their medicines. And so my work is from Axel'One. Axel'One is a collaborative, innovative platform in France, which has been established in 2011. And so the idea of this platform is really to be able to pool together services, tools, and skills to help industrials implement new technologies and make work more efficiently to develop their processes dedicated to chemical production. And especially I work in the platform Process Innovation, and I work on the platform for analytical online analysis. So really bringing our knowledge to help industrials in online analysis. So we've been working for pharmaceutical companies, and there is a shift to go from batch to continuous production. And of course, to do that, we need to implement more and more process analytical technologies, which allows us to better monitor the processes, control the reaction, and develop statistical models to help to do digital twins, for instance, and ensure product quality at the end of the production. So the reaction here in this case that I'm going to be presenting is really based on diastereomer synthesis. And so the idea is that these molecules are very similar, but they have a distinct spatial arrangement, which are not mirror images of each other's, but of course, in terms of similar distinctions, is not always so easy to separate them. So the traditional way of monitoring them is really using HPLC methods, which is a offline analytical method, which is with a high resolution and high accuracy. But of course, it is a lengthy analysis, and the results you get them after bringing the sample to the lab and doing the analysis. So it takes a while. So it's difficult to have a good control loop on your system to be able to adjust operating conditions if you're not happy with what you're having. So the idea is really to test new options for measuring this diastereomer's production. And the idea is to see which technique between all the spectroscopic techniques that exist is going to be the most efficient. And so we have optical spectroscopy techniques that are very easy to implement online. And when you combine them with chemometrics, then you can extract the information you're interested in from these signals, and you can go back to giving the information that you're looking for. So this is what we've decided to do. So about optical and spectroscopic techniques, why do we like working with them? So it's, of course, because it's the interaction between a wavelength, interaction between, I can't find my word, between a material and une onde, a wavelength. And so depending on the energy of this wavelength, you do not scan the same thing in your products. So if you are in infrared or in Raman, you will be looking at the fundamental vibrations of your molecules, of the bonds of your molecules. And of course, if you're doing NIR, then you are going to be starting to look at combinations of these vibrations and harmonics, overtones. And so, of course, it's a bit more difficult to look at, but then the signal is also very stable. So it's usually very interesting to put online near-infrared spectroscopy. But also, if you are lucky enough to have a nice signals in UV, UV can be very interesting because it's very sensitive technique, because we are looking at the transition, electronic transition, which are very intense signals. So, of course, depending on what we want to be looking in the process, I would always recommend to try all of these techniques before deciding to go to infrared or Raman or NIR, because you never actually know with all the constraints and everything what is going to be the best option for you in real life on the process until you've tried it. And so this is what we've tried to do. So about the reaction we are going to be working on today, it's the diastereomer quantification that we want to be doing, the sum of these two diastereomer in the API synthesis. So to do the reaction, it's in fact the reaction we are interested in is really the reaction of an aldehyde with an amine with an acid, carboxylic acid, and isocyanide. So to be able, so this is a Yugi reaction to make the product we are interested in, the diastereomer. But to be able to do this reaction, we actually need to do the first reaction, because we can't, it's not a stable product, the isocyanide. So we need to actually make it in the lab first to be able to do this reaction. So we've actually monitored two reactions, this first step and this second step reaction. So for the first synthesis of the isocyanide, so it's something they already do over there. So they know that infrared is a good way of monitoring online the reaction. So this is what we've implemented. And to check, of course, we were happy with what we were doing. Everything was consistent. We had a low-field benchtop NMR available to do our reference methods on the product. So once we have produced our reactant, which we call reactant number 4 in this case, then we bring this sample to our synthesis batch reactor, which we have implemented with UV, NIR, Raman, and MIR, middle infrared. And so in this reaction, we added all the 4 reactants, and we monitored this product through time. Regularly, we sample some product to be able to collect a fraction and to be able to analyze it with using HPLC for our reference method. once we have done this for several batches, so we get a database, and this database, we can work on it and develop a model to check that we know which signal, how to correlate each signal to the reference values we are interested in. And then we are going to be comparing all of those together. So in terms of instruments that we've been using for this study, so we had a Raman, so from the Indatec company Chauvin-Arnoux, 785 nanometer laser. And the type of probe we were using is a short focal immersed in the product with acquisition times which are fast. In this case, 20 seconds. It's not too short, but it was sufficient anyway for monitoring our processes, and 3 scans. For the MIR, so we had an ABB system equipped with an ATR probe. The NIR, so I'll go forward, is a Brücke a system, and the UV was an Ocean Optics. And of course, they have all specificities, and we somehow managed for all the techniques to have a data every 60 seconds, because this is what we have for the NIR and the MIR, the time we took for each of those. Okay. So in terms of probes, so because this is kind of interesting to understand. So for Raman, Raman is a system where you have inelastic diffusion of the photons that you are collecting. So you can actually send the light through the probe and collect it at the same from the same window as a backscattering. Only you're collecting signals, but it's usually is enough if you collect it right. And so it's a very flat system. The MIR probe is a ATR, attenuated total reflection system. And so you have a crystal on which you do the measurements, and this measurement is really done at the surface of your probe. So you have to have a good renewal of your product every time at the surface of your probe. If you have a reaction with catalysts or with particles and things like that, you will not see this suspension material, because only the liquid film on the surface of the probe will actually have interact with the light. And of course, only this will give you the signature, information about the composition of what you're looking at. If you use NIR, so NIR, you will be working with a transreflection system, because in this case, you can go through a much thicker part of your product. The optical path can be much bigger, and so therefore the light goes forward. There's a mirror at the bottom where you can adjust, in fact, the path length of you want to be measuring in your product, and the signal you're going to get is therefore called transreflection. So it's transmission, but with a bit of reflection signal in it. And then for UV, it is the same kind of probe that we've tested at first. There's a small subtlety here, but we'll see afterwards. So yes, you see already that we have different type of way of bringing the light to the material we want to be measuring, to the medium, and how you collect it also impacts a lot the quality of the signal you're going to get and how you're going to be exploiting it. Okay. So how do we implement all of this? If we look at the picture of what it looks like in our small reactor, so we had an NIR probe in there, the Raman probe, the infrared probe, and the UV probe. There are some small subtleties also. If I take infrared, for instance, infrared, the fiber optics has to be very short. It's only 1.5 meter you get usually, and it's in fact a very rigid fiber optics. It's very fragile, so you have to be very careful when you put infrared online on the system through a fiber optic system. But otherwise, the other instruments, you don't see them in the picture because they can be put much further away. If we go a bit further, so this is actually what the experiment looked like. So the experiment was under the fume hood, and you had the different instruments that can be put further away outside the fume hood on the working table next to it. And of course, there's also all the digital aspect of the problem. So you have to manage all this kind of computers separately because they all have their softwares dedicated to acquiring your signals. So it's a kind of experiment. It does take a while to implement, but it's very powerful. So the nice thing about some of these techniques, contrary typically for infrared, where you had to have the system really close to your reactor, you can see that for others like Raman or NIR, you can have long fiber optics. So in our case, the equipments we get, we always manage to have them so that we can go to the industrial site afterwards to be able to validate all the things that we've done in the lab. And so our instruments are really sometimes the exact same as the one we are going to be doing at the factory plant or the pilot scale plant to validate the final models. So this study here is really just to see the feasibility and select which technique is going to be the most appropriate for modeling. So if I look at first the synthesis of the isocyanide step, so we did monitor this using the infrared reaction, but we needed to check that this was okay, and we did this with fluor NMR. And what we could see is that from the starting material, the product was the two starting materials here, the product was consumed, and at the end only, there was still a bit of the organic stay, but we had produced what we were looking for mainly, so we were very happy. Okay. With that. And if we look at the infrared that we get online for this production, so we can very easily monitor this band, for instance, that shows us the consummation of the reactant amide into transforming into the isocyanide. So of course, we don't go up to the end. You see there's still a bit of it left in the product, but we know at least that we've produced quite a bit of it, looking at the consumption of the amide and not the production of the isocyanide, but that still works. Okay, in terms of the reference method, so of course, when we do this work on the spectroscopy techniques, we also need to develop the reference method. And so in this case, because we are not necessarily specialists in chromatography techniques, we actually benefit from what the industrials already know of their products and how they measure it. And so this is, we've applied a method that they've used, that they've developed in the lab to have the reference method. So we implement the exact same reference method to be able to compare ourselves. And we can very distinctly see the production, what we want to be looking at, so the products we are making, and we want to quantify using the HPLC methods. So that was what we were looking for. In terms of, we always want to check, of course, that we are in good repeatability, because when we want to develop models, the quality of the reference method is also very important, because we cannot hope to make a model which is better than any reference method, usually. And so, in fact, what we did is we established the reference method across all the control points we had done during the student's internship. So she stayed over 6 months. So maybe this control chart covered a period of 4 months. And so this was actually the intensity she got from all the control points she did on our HPLC using the products of interest here. But in fact, also, what she is showing here is that at the end of the product, when the reaction was finished, she managed to do some repetitions of analysis using the HPLC per batch. And you see that you could be, in fact, much better than the control chart. So it's always difficult to have the right estimation of the uncertainty between how to have a good idea, good representativities of uncertainty. And so what we know is that probably the right uncertainty for HPLC is between what we get from the control chart and what we get from the real data from our process, okay, which is apparently a bit better, but it should be in between the two. Okay. So now, if we look at the results for the spectroscopy techniques, so these are the spectra we got after analyzing the Raman. So the Raman, very quickly online, we started to see a very strong fluorescence issue with the background going up, up, up. And so in fact, we hadn't noticed it early on. And because we did not have the isocyanide molecule available to check that Raman would make sense or not. And unfortunately, when we actually monitored the reaction with the isocyanide, well, then we saw that we had a strong fluorescence. So unfortunately, this technique proved inefficient, and we had to give up on Raman spectroscopy for monitoring these processes. So in fact, we had done the pure compounds we had available. It would seem that it should have worked, but in fact, when we were inside the reaction, with everything present, we discovered there was a fluorescence. So that unfortunately happens quite a few times with Raman. You think it's going to work, and then when you're live on the product, well, you discover there's traces or large fractions of compound that fluoresces and prevents you to do good monitoring of your process most of the time. One option would have been to monitor using fluorescence technique, which are becoming available on the market more and more. But in this case, we haven't done it, especially with the problem with fluorescence. If we want to monitor using fluorescence, it's a very non-linear technique. And so the chemometrics behind is going to be more complex. And well, we need more feedback, I think, for being comfortable in using fluorescence for monitoring processes yet. So now, if we look for UV results, so UV, we had very nice signals for the pure compounds UV. So that would seem it was very interesting for the product, for the reactant 2, reactant 3, for the aldehyde here, reactant 1, and for the solvent. So very distinct signature all the time. So of course they are overlapping, but with a bit of chemometrics, we thought, okay, this could be a good technique. Unfortunately, when we had the isocyanide again, we thought we had a very strong signal from isocyanide. And so everything we had done offline before going online with the pure compounds, we had done with a transmission probe, a transreflection probe, sorry, with a small path length of 2 millimeters, it completely saturated. So we, in fact, had the opportunity of going to HER UV technique with a katana from Hellma probe. And of course, with HER, the optical path is really small. It's kind of a few microns, depending on the wavelength you're using. And so this was actually perfect. This allowed us to have still signals. Maybe you can't optimize, of course, the optical path for your product, but if you still have signals, then you can work from that. So we shifted for online from Falcata to the Katana probe for UV. And when we look at the spectra we got for monitoring our batches, then we see we had very nice signatures, which would evolve in time. And so that was kind of interesting to see, especially here in this case, you see how for one batches it evolves after pretreatment. So the pretreatment we used was working on a smaller range from 200 to 350 nanometers with a derivative that allows to compensate for very fluctuating baseline issues that we would have, as you can see, depending on the different batches. And yes, well, that was very interesting. And you could also see that here in this case, it's colored according to the what we want to be predicting. And so that so the sum of diastereomers. And so you see you have very nice color gradients in your data. And this color gradient is really the good sign that you are going to be able to monitor what you want with the signals you are collecting. So that's what we did. We did our models using PLS. And this is the parity plot of your models with the calibration in gray and the validation test. No, so it's not validation, it's test. I believe in this case, yes, it is. It is a test data set. So a few of the batches were taken away from the calibration base to be able to predict them, test them later on. So this is the result of that work. Okay, and so and in this last graph, you have the distribution of residues showing that it's very evenly distributed along the zero line. So there's no strong nonlinearities or bias in our model. So this was the result for the UV, and this is the model we did for the mid-infrared. So for the mid-infrared monitoring, what happens is that it was not so easy to develop the model in this case, and one way of actually did help enormously in how to deal with that was to do variable selection. And so the student actually discovered this because I was not a specialist, but it's an option that really helps you to do this variable selection in the model. So we use the software which is called PLS Toolbox or Solo from Eigenvector. And using these options, we actually managed to do a pretty good model for the mid-infrared from the mid-infrared data. So for which we were kind of happy. So root mean square error of 0.18. And if I go back, sorry, to the one for UV, we had a root mean square error of 0.16. So very similar values you will see for all the data sets. So yes, maybe what I did not say is that we did test different batches during the process to be able to collect enough data. And of course, from one batch to the other batch, you see there is a lot of variability. You see it especially with the NIR spectroscopy. You have a very distinct baselines because even though you think you start from the same thing, optically, there are often a bit of scattering, especially for NIR or different aspects that's you will not be able to compensate so well when you are online on the process. There is a source of variability which you cannot control so well. The way of controlling it is really using the pretreatment as we are doing here. Again, a derivative, which I personally really like when I'm doing online analysis because it compensates also for fluctuations maybe of temperature we don't control so well and different things. And it really helps to make a good sense of all this data that was collected, which has a strong variability, and really make it disappear so that we can concentrate on the chemical information left in the spectras. And here again, you see very well that if I color the spectras, the information according to what I want to predict, we have very nice gradients. So for sure, the information is in our spectras. So in this case, when we developed the model, I think there were still a few batches that we could not integrate in the calibration. So these models, if I compare them, they are based on a slightly less data than with the other techniques. Yes, that happens when you have 4 probes. It doesn't mean that every signal is going to be you're going to be exploit them. You have different causes of having problems with the acquisitions and stuff like that. And so you would probably have to check every time all of your signals to check what is reliable enough for you to build your calibration on. Okay, not everything is always completely safe. And so in this case, if I want to compare the root mean square error of calibrations, for instance, you see it's a bit smaller than for the other techniques. It's 0.12, where before we were at 0.16 and 18, if I remember well. And this is probably due because we have a bit of less data and distribution, so errors, causes for errors in our model. So it inflates a bit the results. Okay. And again, here, if I always look at the distribution of my errors for what we call the heteroscedasticity of the errors, if it's well distributed along our zero line. And here, as you can see, it is pretty nice. Okay. So now, in fact, when we do online analysis, the models are very nice, but what you really want to check is how good is the monitoring I'm doing with these models at the end. And so this is what is shown in this slide, and it's probably the most important one when you need to decide which technique you want to be implementing online is how good is my monitoring at the end. So when you are online on a process, it's very important to make a model which can be as robust and reliable, depend on not too many information, so that it can still work from one batch to the next. Because it's really difficult to acquire all the variability that you will get. There's always something happening which make the spectras not as stable as in a lab process. So the chemometrics you are doing when you are doing exploiting online data has really to be much more simplified as possible. You don't want to put too much information in there, otherwise your models, they're going to react badly. So overfitting is our worst enemy. And if we want to compare, so how we predict using the different methods compared to our HPLC reference method, so and its uncertainty, so we're all, more or less all of them are in there, but do they talk to me about what I really want to see in my models? And if I look at the monitoring here, it is the monitoring for the MIR, which was done using the online data and the models that were established. So we see that it does follow the trend really pretty well. There is a bit of overestimation, for instance, for this batch, especially, but otherwise it does work. But what I don't like is the noise. You have a monitoring which is really noisy. And in fact, what you want to do is to be able to know if I go up, is it significant or not? And in this case, it's a bit so noisy that is difficult. And this is really typical of unfortunately ATR. Anyway, our ATR, it is the signal to noise ratio is not as good as you would hope for, especially maybe because in our case, so our ABB system is a DTGS, because we want to be able to take our system on the industrial plants where there is no liquid nitrogen, for instance. So we really needed this DTGS detector, which is maybe a bit less sensitive than the classical MCT detectors. But so this maybe is why our signal to noise ratio in our spectra is maybe not so good. Also, the model, as you remember well, was much more difficult to develop. We had to do some very intense variable selection. So yeah, maybe this would explain why our model is not probably the best in this case. But anyway, infrared online is a bit of a risk, especially because you have such a thin optical path and the risk of fouling is so big, which makes it a bit tricky to want to implement online. If I take the UV-Vis monitoring process, so, well, this talks for itself. It looks really, really nice. If I look how it fits the HPLC with my UV, I can see the little, if with the blue lines, I can really see even a small inflection that maybe I would have missed if I had only the HPLC values there. What's interesting also is what happens at the beginning here. So this is the beginning. So it's I put it in gray. It's because we are in fact starting the acquisition of the spectrals even before the reactions really start. So all of these, we are predicting values for the sum, but of course it doesn't make any sense because we're not in the conditions of the reaction yet. But what we can see is that even with this, the signal is already telling us a lot of stuff about what's happening while you are introducing your product. For instance, here, when we start introducing the reactant number 3, or anyway, the reactant we introduced at this time, we see that it was starting to evolve. So the introduction and some kind of reaction already is starting. So yes, what I was showing is really this region where once you've introduced your product, you see something is already happening in the reaction. So that I think is really interesting with the UV, even though the model is not appropriate for this part of the process because it's not what the model was built on. It's already saying something about how the system is already reacting when you start introducing this product. And so this is for the UV. And now if I look last for the NIR monitoring, so the NIR also for this part, well, you see, I'm not so happy with the NIR monitoring in this case because it is showing very strongly maybe an inflection point here, but it doesn't correspond exactly to what I wanted the model to say. I really wanted it to fit with the HPLC values, and in this case, it's not so much the case. In fact, this is a bit of a problem with NIR often, is that NIR is very susceptible to a lot of things in the environment. It's a combinations and overtones, so it is much more susceptible to interactions of your molecule with whatever is happening outside, temperature or whatever. And so this is the models are much more difficult to make them very specific to a specific compounds, because it will be susceptible to all kinds of process condition variations and things like that. And even though we made a model we were very happy with, we still have a bit of issues in how good the fit is compared to the HPLC, which we wanted it to fit to.
Noémie?
Yes?
I think now the people can't see the pointer, but it's okay.
Oh, anyway, and I've more or less finished because I'm with the conclusions now. So the idea is was just to say all the techniques showed that you could develop models which were rather good for being able to monitor the process. In terms of root mean square error of calibration, of course, we only did it with a few batches. So I think in this, it was 7, or I don't exactly remember, but there was only a few batches that were really used for the calibrations models. So of course, we could add more to make better models always, more representative and things like that. But still, it gives you a good a feeling of which technique allows you to get a better sense of what you want to be analyzing. And so in terms of uncertainty, values were comparable to the HPLC reference methods. So in fact, I think that all of these techniques could have a good indication of this, but definitely, if I had to recommend a technique, in this case, it would be UV specifically, because in fact, it's a cheaper spectroscopy usually compared to the others, and very easy to implement and rather specific to what we wanted to measure in this case. So this is a good thing because spectroscopy is the difficult thing is having the specificity to what you want to be measuring, and so UV definitely is a good thing. And the combination of UV with ATR UV it's not often I've been able to find a good application for it, but in this case, it was a very nice combination of this process with the ATR UV. Okay. And yes, the idea would be always for these studies, once you need to re-implement the model once you are in the real conditions of the process, but at least the first model we got from the lab is a good first trial to model and to monitor your process, even though you might be biased compared to what you're going to get on the industrial site. You have a good grade of how to read your spectra and get a good semi-quantitative approach to what you want to be measuring for future tests. Okay, thank you very much. I hope it's been clear, and I would be happy to answer any of your questions.
Thank you very much, Noémie.
Okay, my pleasure.
So if you want to share your PAD experience with us the next time and be the next speaker in our PAD forum, feel free to contact us via email. We were very happy to have you as the next speaker in your next PAD forum. So thank you very much for the presentation, Noémie. And thanks to our participants. So we hope to see you again the next time for another exciting PAD topic. And please find our upcoming webinar dates in a good time in advance in our Hellma website. So have a great day, and yeah, merci Noémie. Bye-bye.
Bye-bye.